All right, today we're tackling one of the biggest stories out there. I mean, the story of how life itself works. We're going to go on this incredible journey from a tiny blueprint inside your cells all the way to the finished product, which is, well, you. So let's dive in. I mean, have you ever really stopped to think about it? How does a single set of instructions build everything that makes you, you? Your hair color, your height, how you digest your food, it all comes from this one single master plan. So how? How does a plan on a microscopic level get turned into, well, a person? To figure that out, we've got to zoom in, like way in. Okay, so our journey starts right here, inside one single cell. You're made of trillions of these things, right? They're the basic building blocks of, well, everything. But the instructions we're after, the master plan, oh, it's not just lying around. It's locked up tight in a kind of high security vault. And as we go deeper, we find it, the nucleus. This is basically the cell's brain, the command center. And inside that is the whole shebang, the entire genome. We're talking about a set of instructions so huge that if you printed it out, you'd have a library of thousands of books. It's just massive. So you can't just have a library's worth of information floating around, right? It needs to be organized. And that's where chromosomes come in. Inside the nucleus, all that information is neatly packaged into these little structures. For us humans, we've got 23 pairs. So if the genome is the encyclopedia, then each chromosome is like one of the volumes. See? Super organized. Now, if we could grab one of those volumes, one of those chromosomes, and carefully unwind it, we'd find the star of the show. You know what I'm talking about. DNA, that famous double helix. This right here, this is it. It's the physical molecule that holds the code, the actual blueprint for literally everything the cell will ever do or build. Okay, so we're not about to read the entire encyclopedia today. That would take forever. Instead, we're going to focus on just one page, one single recipe. And that's what a gene is. It's just a specific chunk of DNA that has the instructions for making one very specific thing, a protein. All right, so think about it. We've got our recipe, our gene, but it's locked up safe and sound in the nucleus. The problem is, the factory where we need to build this thing is outside the nucleus. So how do you get the instructions from the vault to the factory floor without risking the original master copy? You send a messenger, and that brings us to the first big step in this whole process. So whenever your body decides it needs a certain protein, that specific gene gets switched on. It's like a signal goes out. And that signal calls in this amazing little molecular machine, an enzyme called RNA polymerase. Its job is simple but so important. Make a temporary, disposable copy of the instructions from that gene. This whole copying process is called transcription, and it's incredibly cool. The RNA polymerase just latches on to the start of the gene, and then it just zips right along the DNA strand, reading the code as it goes. And as it reads, it builds a new strand, a copy, called messenger RNA, or mRNA for short. But wait, it gets better. Before the message is sent out, it gets a little edit. Any bits that aren't needed are snipped out, just to make sure the final instructions are absolutely perfect. So now, with its marching orders finalized, our little RNA messenger leaves the safety of the nucleus. It travels out into the big, bustling main part of the cell, which we call the cytoplasm. This is where all the action is. This is where the factory is waiting to get to work. Okay, the messenger has arrived. Mission accomplished. For part 1, anyway. Now it's time for Act 2, translation. This is where we take that coded message, which is written in the language of genes, and translate it into the physical language of proteins. Let's head into the factory. So the first thing that happens out here in the cytoplasm is our mRNA message gets snagged by this thing called a ribosome. And you can totally think of the ribosome as the factory itself, or maybe the main assembly line. Its whole job is to read the message and start building. Now, every factory needs raw materials, right? For this one, the materials are called amino acids. Get this, there are 20 different types of them. And the ribosome's job is to string these amino acids together, kind of like beads on a string. But here's the absolutely critical part. The order matters. The specific sequence of those 20 building blocks is what determines everything. It's the whole ballgame. So you might be wondering, how do the right amino acids show up at the right time? I mean, it has to be precise. Well, that's where another player comes in, transfer RNA, or tRNA. Think of each tRNA molecule as a little delivery truck. Each one is specialized to carry just one of the 20 types of amino acids, and it brings it right to the ribosome when it's needed. Okay, so here's how the assembly line actually works. The ribosome clamps down on the mRNA message and starts reading the code, but it doesn't read one letter at a time. It reads the letters in groups of 3. These triplets are called codons, and each 3-letter codon is the code for one specific amino acid. So the ribosome reads a codon, the right tRNA delivery truck pulls up with the matching amino acid, and boom, it gets added to the chain. It's that simple, and it happens over and over.
And the ribosome just keeps chugging along that mRNA strand, reading codon after codon, adding amino acid after amino acid, just building this chain at an incredible speed. It keeps going until it hits a specific triplet that basically says, stop! And that's it. The chain is built. The basic construction is done. But believe it or not, the most amazing part is still to come.
Because this is the final, absolutely crucial step. That long chain of amino acids, it doesn't just stay as some floppy noodle. Nope. Instantly, I mean in a flash, it folds. It collapses on itself, driven by all the little chemical pushes and pulls between the amino acids, and it forms this incredibly complex, precise 3D shape. And this isn't random. It's like pre-programmed origami on a molecular scale. It's just mind-blowing. And that's it. That folded 3D shape, that is the protein. Its function, what it does, is all determined by that shape. Whether it's an enzyme that makes a chemical reaction happen faster, or a little brick that helps build part of the cell, its job comes from its shape. And just like that, from a simple line of code back in the DNA, we've created a brand new working protein. Now, get this whole incredible journey we just took, from gene to protein, it's happening in your body right now. Billions, trillions of times, every single second, with almost perfect precision. It is quite literally what life is, which kind of leaves us with one last big question to think about, doesn't it? If this entire unbelievably complex process depends on getting that code exactly right, what happens when just one of those letters is wrong?